Hello, so today's episode is going to be about the steps in thyroid formation. So once we discuss this in detail, it'll be much easier for you to understand which are the drugs affecting which part of the whole cycle. Okay, so you can see over here, this is the blood vessel. You have the thyroid follicle cells, and inside this is the follicular fluid which is there. Okay, now your iodide is usually absorbed from your GIT. Once it is absorbed, it reaches the thyroid through the blood vessel. So the first step is for iodide to get into the follicular cell. This is what is called iodide trapping. So this involves a sodium and an iodide co-transporter. So two sodium and one iodide. So that is how it is transported inside. But this requires energy. So this is a sort of active transport. What type of active transport? So for this to take place, actively sodium is pumped out and potassium is taken in, your sodium potassium ATPase, and this creates a gradient for sodium to come in, and when sodium comes in, along with that iodide comes in. So this is the type of secondary active transport. Okay, so it's a type of secondary active transport. So this step is called iodination, or it is called iodide trap. This is how iodide is trapped into the follicular cell. Once this is done, next this iodide has to be taken into the lumen, okay, where the follicular fluid is there. So for that, there is a transporter which exchanges it with chloride. So iodide, chloride is exchanged, and this is what is called Pendrin. So this Pendrin transporter exchanges chloride with iodide. Now, apart from this, you can see that, it's all the same cell. Okay, so you can see the nucleus, the endoplasmic reticulum, the Golgi. So thyroglobulin, through transcription, translation, and the whole packing, it is processed and stored in secretory vesicles, and it is released inside. Okay, so when this is released, this thyroglobulin, the secretory vesicle containing thyroglobulin, not only has thyroglobulin, also has peroxidase. Peroxidase is a membrane protein, integral membrane protein. So it's actually not completely released outside. Instead, it goes and attaches to the membrane. Okay, so this structure is an integral membrane protein called the peroxidase. So this apically, when it is abutted into the membrane, this results in activity. So you already mentioned how through Pendrin, iodide has come into the fluid, and this will now be activated upon by your peroxidase. So this will convert it into your oxidized iodide and your nascent iodine. So oxidized iodide and nascent iodine. So for this, you require H2O2 peroxide. Okay, so this step that we are talking about, this is oxidation, and as I had mentioned earlier, it requires peroxide. Okay, so this peroxide is provided by another enzyme. So that is a dual oxidase 2. Okay, so after iodide is trapped, it goes out through the Pendrin and it is oxidized by peroxidase. So this step is oxidation. That requires a peroxide, and that is provided by dual oxidase 2. So now it gets very complicated, everything's up here. So just remove it and we'll go to the next step. Okay, so now this thyroglobulin is also out here. So thyroglobulin, you have to understand, has tyrosine residues. Amino acid tyrosine residues are present. It has around 70 to 100. Some textbook even say 50 to 100. So that many different tyrosine residues are there. So your nascent iodine and your oxidized iodide, they are now going to go and iodinate. So this is what is called organification. Okay, so the tyrosine residue on the thyroglobulin gets organified. Let's just take a look at what happens over there. So you can see your tyrosine structure. This is attached to your thyroglobulin. So we have OH cycle over here, CH2CH and H2COH. So this is your tyrosine, which is attached to your thyroglobulin. Now, when organification takes place, this going and one iodide coming over here, this is what is called monoiodotyrosine. So only one iodide is attached over here. So that is monoiodotyrosine, MIT. Similarly, if this goes and attaches in two locations like this, so this becomes diiodotyrosine, DIT. So I hope this is clear, very simple. Now we'll come to understand what coupling is. So till now we had iodide trapping, through Pendrin it's gone out, and then oxidation took place, and after that, along with the thyroglobulin, that oxidized part is now going to go organify. Once organified, we have MIT and DIT. So this MIT and DIT in different combinations will be coupled together. Okay, so that is your simple coupling. So we have your MIT, as you can see over here, one iodide, and this is the structure. This MIT with a DIT. You can see two iodide. So this is MIT with a DIT. So this part over here, you can see that H is actually this side. So OH and CH2, CH and H2, COH. So this H is transferred here, and CH3, CH and H2, COH goes outside. So that finally forms a bond like this. Okay, so this is actually a addition of MIT along with DIT. So MIT and DIT, it forms 1, 2, 3. 3 iodide residues are there. So that is your triiodothyroxine. I hope this is clear. So this is how triiodothyroxine is formed, MIT along with DIT. On the other hand, if DIT is there along with DIT, okay, this is pretty simple. So similar way, 1, 2, 3, 4. So there is 4 over here. So earlier it was T3. Okay, now T4 is formed, and this T4 is what is called thyroxine. Okay, so this is your thyroxine and T3, triiodotyrosine. Now, this structure that you see over here, this is MIT with DIT. This is your T3, regular T3. Okay, instead, let's try something else. We'll take this DIT and then add it to MIT. So now, when this whole thing happens, it's going to come like this. The O is going to come over here. Okay, so this is DIT plus MIT. Okay, so this you can see is actually the whole thing reversed. This OH is over here in the MIT region, and the CH2, CH and H2, COH is at the DIT region over here. Instead, now it's reversed. Okay, so this is what is called a reverse T3, RT3. Now, just to recap, MIT plus DIT is your T3. DIT plus MIT is your reverse T3. And DIT is your T4. Okay, your thyroglobulin has tyrosine residues, and along with it, after organification, we have MIT, DIT. So some will be MIT. I remember there are like 70 to 100 residues. So there's MIT, there's DIT. There's also T3, T4 after coupling and all that. So everything together is there in this, on this thyroglobulin. Okay, and this gets stored in the follicle. It can get stored up to 2 to 3 months inside the follicle. And later, as and when required, it's taken. Okay, so this uptake is based on a fluid phase endocytosis. Okay, it's a type of pinocytosis. So in case you have a doubt regarding what this is, I already put up a video describing different transport mechanism. So just check out that link above. So like this, fluid phase endocytosis is taken inside, and once inside, it's there inside a vesicle inside the follicular cell. Now lysosomes degranulate into it, and it results in, the enzymes inside the lysosome will result in proteolysis. Okay, because of that, T3, T4 is removed and it goes out, and finally it's taken out into the circulation. MIT, DIT is also removed out. So this T3, T4, once it is there in the circulation, it cannot directly go through the circulation like that. It needs something to bind to it. So this is bound to thyroid binding globulin or albumin or transthyretin, also called prealbumin. So thyroid binding globulin, albumin, transthyretin. So attached to them is how your T3 and T4 is going to go into the circulation. This MIT and DIT, on the other hand, by action of deiodinase, they get converted back to iodide and is taken back, and the whole cycle repeats itself. Okay, so now that all this is done, let's just recap through everything. Okay, so first of all, iodide coming from the GIT gets iodide trapped. Okay, so iodination takes place, and that requires your sodium iodide co-transport, secondary active transport. Then with the help of Pendrin, it's going to go out into the follicular fluid, where with the help of peroxidase, oxidation takes place. Nascent iodine and oxidized iodide. So this, along with the thyroglobulin that is going to come out, okay, so this goes to organify it on the tyrosine residue. And once MIT DIT form, they are coupled together. Okay, and that coupling will form T3, T4, reverse T3 and all that. Now after that, after storing up to 2 to 3 months, whenever it is required, it is taken back. So that requires a fluid phase endocytosis through uptake, and your proteolysis step takes place because of lysosome enzymes, and it is finally released into the circulation. So once this thing is understood, we'll try to understand which are the drugs affecting various steps over here. So first is your iodination. Okay, it's like a very important step in this. Thiocyanate is something that affects this, inhibits this. Also, perchlorate is something that inhibits this. So perchlorate, we earlier thought that it was sort of like a competitive inhibition. So perchlorate actually is known to actually move through this in 1:1 ratio sodium along with perchlorate. Okay, it doesn't actually inhibit it, but because that also comes into the amount of iodine coming through it is reduced, that's all. Okay, so thiocyanate, perchlorate. Also, this is very important, increased, very high concentration, not increased, very high concentration of iodide. So very high concentration of iodide will inhibit the synthesis of thyroid hormone. Okay, so this is what is called Wolff-Chaikoff effect. So this is one way it acts. Other than that, it also affects this. So after the coupling, for it to be taken, so this uptake, proteolysis, release of hormone, this step is also affected. Okay, so just note, it's actually the uptake that is affected. Because of that, ultimately release of hormone is also affected. So Wolff-Chaikoff effect, very high concentration of iodide will affect iodide trapping as well as your uptake into the follicular and release of the hormone steps. Apart from that, we have the Pendrin. So a defect in that is seen in a syndrome called Pendrin syndrome. Okay, so this has a series of things, even sensorineural hearing loss, things like that are also there in this. Okay, so that is your Pendrin syndrome. Then it goes into the follicular fluid. Oxidation is affected by propylthiouracil. Now organification is affected by your methimazole, and coupling is affected by carbimazole. So propylthiouracil for oxidation, coupling carbimazole. Okay, so next is TSH. We have to understand when TSH acts, what are the steps that are improved because of the presence of TSH. So first and foremost, your iodination is improved. Okay, so because of the presence of TSH, the more number of these transporters are released. Okay, because of that, iodide trapping increases. Also, the thyroglobulin synthesis and release is also increased by TSH. And your whole step of uptake, proteolysis, release, this is also improved by TSH. So TSH, almost every step is affected. More importantly is your iodination, iodide trapping, the release of thyroglobulin, your uptake, proteolysis, along with the release of hormone. So I hope this was helpful in order to make this much more simpler and easier to remember. Thank you.